Okay, so I had this idea last week, and I was sort of noodling over it. I ran it by briefly by Marcus on Friday just to double check on this practice it. And I'm going to try to do it very quickly here. And it might, as often, sometimes in the beginning of these things, it's very difficult for me to figure out how to describe it. So hopefully I'll do a reasonable job, but I wouldn't be surprised if I don't. But it's a potentially very, very interesting idea. So I'm going to go through 1, 2, 3, 4. I'm going to start off with some basic background just to remind some stuff what Mountcastle proposed. He proposed that the mini columns were the unit of replication of the cortex, not columns, but mini columns. And that idea came from that if this is a section of cortex, there's these progenitor cells that during development, they grow and they send out a series of neurons that populate a very thin vertical column. And there's about 120 cells that's reported in each of those mini columns, and they contain all functional types of neurons. So the idea is that there aren't multiple types of mini columns, that each mini column has a complete set of cell types. If you just do the basic math, it would be about 150 million of these mini columns total in your cortex. And then he defined, he said that's the unit of replication, but the functional unit is the larger column. And that is not a physical thing. The mini columns are physical, but the functional unit is just sort of a thing that's built up of several hundred mini columns. And the way you might think of it like that is if I had a sensory region, and this is my sensory array of whether touch or vision, and you have all these sensory bits coming in from here. Well, imagine they're coming up in a topological fashion. Within a larger column, they're mixed together. So there's no topology in this larger column. Every mini column in that larger column is potentially connected to anything down here. When you go over to the next larger column, that's representing a different area on the sensory array. And so he showed these papers that as you move across through the cortex, you have this like all right here, that it jumps all over here. We reviewed those papers. Okay, I'm going to talk about the mini columns here today. He proposes that we've proposed a partial explanation of what mini columns might be used for in our sequence memory. There are very few people who have theories about the mini column, and I'm going to propose one here. The second observation, this came from Hubel and Wiesel, and now lots of people, there's many, many papers on this idea, but what they found is that many of the cells, and about 50%, it's not everyone, in the mini column share receptor field properties. And so as you go across all the layers, there's commonality there. Now, we think about it, actually, often, I've made a mistake, think about it often as some sort of spatial property, like, oh, there's an orientation in it. But what they actually found, and what they reported, that most of these cells are directionally sensitive. The vast majority of the cells in the column are directionally sensitive. They care about which way the sensory movement is occurring, and they're graded in their response in that regard. I've always wondered about this because I said, why would so many cells be directionally sensitive? It's sort of like, well, maybe that's like I'm trying to recognize something that's moving. I'm using some sort of temporal inference. I'm going to propose a different answer to that right now. Another observation just worth mentioning is that in the upper layers, in some sense, I'll just leave it poorly defined, but receptor field in the upper layers is smaller than the lower layers. If you look at what the cells in the upper layers respond to, they tend to be shorter and smaller items, and the lower layers are much bigger. That's another observation that was sort of pulled out back a long time ago. So that's some background information. Now, we have jump over here. We proposed the thousand theories of brain, the thousand brains theory of intelligence. And what we said is every larger column is a complete sensor motor modeling system. And therefore, every column, the larger column, this means the word column means larger column, I put M column for the mini column. Every column must have a multidimensional metric space. We've talked about that a lot here. The only examples we know about of metric spaces are grid cells from the entorhinal cortex. Therefore, we conclude that there must be multiple grid cell-like modules in a column. One grid cell module isn't efficient. You need multiple ones to define a space. So that's one of the premises of the thousand brain theory of intelligence. There also has to be a multidimensional orientation, match the multidimensional metric space. So we have to know which way, if you're a person, which way you're facing, if you're a finger, which way your orientation, and so on. And therefore, there has to be multiple cells that represent orientation relative to the space defined by the grid cells. So this is part and parcel of this basic stuff that's in the theory of thousand brain theory question. tells us. There's a lot of things we don't know. These are some of the puzzling questions we've had. For example, where are the grid cell modules in a column? We think they're in layer 6, and we do that by the connectivity to layer 4, but we actually need many grid cell modules. It's not obvious how you fit many grid cell modules into layer 6. We've had the same problem in the entorhinal cortex. We've looked at the entorhinal cortex and the research on that, and people show, oh, there's a grid cell module. So we need many of them to actually, multiple and lock, if we could, to make it work. And it's not even clear how it works in the entorhinal cortex. It's not clear how it works in the cortex. Next question is, what is the dimensionality of the grid cell modules? We think of them as 2 dimensions in the entorhinal cortex, and maybe that's true, maybe it's not. We've read a whole bunch of papers about how some animals might map into 3 dimensions. It's not clear what it is in the cortex. It could be 2 dimensions, we don't know that. We don't know where the orientation cell is in the neocortex. And more importantly, the orientation and the grid cells have to work together. Like how you update the grid cells depends on your orientation. If you go in one way, the grid cells modify one way. If you go a different direction, they modify another way. And that has never been really clear to me how that happens. There's been some theories about this in the entorhinal cortex, but that's kind of a mystery. It's problematic how that works. Then there's another thing we talked about here. We think about grid cells being motor derived, but we concluded that grid cells have to be updated purely by sensation as well. Remember that this was the idea that you're sitting playing a video game, and as you're moving through the video game, you're not actually walking, but your sense of location changes. And so that has to be based on flow information on screen. And then I think we read a paper which talked about something about this too, but the point is that grid cells need to be updated by sensation. We know that has to happen. We also know that grid cells need to be updated by movement, but I'm going to argue that I always felt that this had to be learned. That is a column, it's getting some sort of motor input, which I haven't shown here, and that motor input can move we know that can move the bulk of the grid cells. But I always felt that had to be learned, that a column doesn't know what motor behavior is. It has to somehow figure that out, how to do that. So I'm going to focus on the sensation part of this talk. And then there's also the question, how do grid cells and orientation cells get anchored to the sensed object? Like we just say, when you come into a new room, your head direction cells just anchor to the room the same way every time. And the grid cells anchor to the room the same way. I don't think we've ever had an explanation for how that happens. How does it know how to anchor again? So these are a lot of very puzzling problems, and I'm going to suggest a solution to them. The solution goes like this. I'm calling this an idea, maybe a proposal. I'm not ready to call it a proposal yet. It's just an idea, but it's a very exciting idea for me, and I'm not sure if it's right or not. The basic idea, I said I have to answer these questions, and the basic idea goes as follows. Each mini-column represents a one-dimensional sort of system. It's representing one dimension in the space that's being modeled. So a set of mini-columns is an over-complete basis set of dimensions. So I might have 100 mini-columns. I may have 3 dimensions in physical space. I'm going to disperse my one-dimensional vectors in an over-complete fashion, and that's how it's going to work. A set of mini-columns can represent any number of dimensions. Meaning it can discover, I'm going to argue that it can discover the dimensionality of spaces. If it's a 2-dimensional space, it'll end up putting many columns pointed in 2 dimensions around 1 dimensions in a plane. If it's 3 dimensions, it'll do it in 3 dimensions. If it's 4 dimensions, it'll do it in 4 dimensions. It doesn't really care. And it's going to learn, it's going to figure out, based on its sensory input, what the dimensionality of the space is. So you can imagine these one-dimensional grid cell modules that are pointing in different directions. If you got a question on this, let me know. This is the key thing here. And then I'm going to say that each mini-column is a 1D grid cell module, and it's going to be in layer 6. Now I'm going to show that over here in a moment. I have no physical evidence for this, okay? There's none. I'm making this up. We do think we have a lot of evidence that grid cells are in layer 6. I won't review that evidence right now, but I'm going to argue the following. Imagine these are mini-columns. And there's a bunch of cells, maybe in layer 6A or something like that. There may be 20 of them. I don't know how many. It's like hundreds. It's obviously this, but it could be 20 or something like that. And I'm going to argue that there's a bump of activity in that mini-column, and that bump of activity moves up and it moves between cells. It doesn't have to physically move up and down. It could, All I'm saying is that that you can think of the cells here as a grid cell module in one dimension. And there might be a couple of bumps so that it can wrap around easily. I don't really know. But the point is that each of these mini columns has a bump that represents a grid cell-like repeated location in that one dimension. And if I wanted to read out my current location, so if I have hundreds of these guys, I can just subsample from them almost randomly, and I'll be guaranteed to get a good representation of location. These bumps have to move when you move, right? That's the whole idea. Is if I'm moving my finger over an object, or my eyes are moving, looking at different points, or I'm walking through space, these have to move, but they don't all move at the same rate. They have to move depending on their orientation. So if I'm a grid cell, a mini column that's representing this dimension in this room, and I walk in this direction, well, that bump has to move. If I walk in this direction orthogonal to it, the bump doesn't move at all. If I walk in this direction, it's going to move partially in between. So these guys are going to move at different rates depending on the projection of the three-dimensional or n-dimensional space onto that one-dimensional vector.
But each one's going to move up and down. The bumps are going to move up and down. Yes, each one. Well, some may not move if they're orthogonal at any point in time. Now, for that to happen, so there has to be some sort of scalar representing how much of these should move, and I'm going to propose how that comes about. But you can think of it. I'm sort of showing that right here. You can imagine like that if I'm not moving, there would be some scalar that says nobody's supposed to move. And if I am moving, there's going to be something like, well, these guys here are more aligned with the direction of movement, and these are not aligned with the direction of movement, and therefore these in the middle will be changing faster, and these on the side will be changing slower. But this is in allocentric coordinates. This is in the object's coordinates.
Yes.
So the movement, how fast it changes, depends on the scale of the object.
Well, it depends on.
It's like a given movement might be completely different scale in different objects.
Well, if I think of it just as a metric space, if I think of it just as a metric space, then and the metric space is fixed in size, then there wouldn't be a scale involved. I could just say I'm defining some space around some object, and I'm not dealing yet with like how would I deal with a small object or a big object. If I said here's a coffee cup, and I'm going to put space around it, there's no scale to that. The space is just whatever it is, and then I attach the cup to that space, right? It's. I guess I'm saying I'm not sure if I agree with you. There's a scale or
right. But if you make a certain movement, these things are going to update at a certain rate.
Yes.
Now, if you take your marker and you have a location space around that, and you make the same movement, is it going to update at the same rate, or is it going to?
At the moment, let's say it's going to update at the same rate. It's like this is physical movement on my finger, if you want to think of it that way. So if my finger moves 3 inches, these columns doesn't matter what they're representing.
Then I don't see how it can be an object-centered coordinates.
Why?
I guess you could say the scale is the same for every object.
Well, imagine for this particular module or this column in the cortex, it defines a metric space of a certain size. And it's going to try to model everything within that metric space, just like grid cells in the entorhinal cortex. I don't think.
But like think about this coffee cup in my eyes, and I'm making a certain saccade from here to here. Okay. If it's further away, that same saccade would go.
So let's leave that out for the moment.
Okay.
Let's just talk about like my finger touching the object. All right. Like I don't have a complete answer.
So we want to leave the scaling question out of it.
And for the moment, I'll come back to it in a moment down here.
Okay.
Right.
It's still a metric space in the sense that you can do computations on things like distances and whatnot, but it's not a metric space in the sense of having an absolute scale.
Well, at this moment, I'm describing.
At this moment, the scale is the movement. In some sense.
Think about when we think about grid cells in the entorhinal cortex, right? No one says, "Hey, those grid cells, the spacing depends on the size of the room." Their spacing is their spacing. And that's kind of what it is. And so there's different grid cell modules that have different, they have different space for the different grid cell modules. So if I have a really big room, one grid cell module would be better at it. And if I have a really small room, another grid cell module would be better at it, right? But there's not like one grid cell module that's moving like this.
It's just a reservoir computing idea. You have a lot of these, and there's going to be one appropriate scale.
Yeah, the problem is we need multiple ones at the appropriate scale.
because one appropriate scale doesn't give you a location.
No, it's not like reservoir. Each one has a random scale and random direction. And all of them together will give you the location. And
I'm not saying that these are random scales. Certainly, there's no point in having one have a very large scale and another small scale. I've made that argument before.
but they're not tied. One mini column is not tied to the scale of the other.
but they would be similar in some regards.
Okay.
The key thing here is they're all pointing in different directions.
Right.
That's the key point. It wouldn't make sense for this to have a scale that's 5 times larger than this one because I've pointed out before, then it would be difficult to read out a location here because you'd be biased by the cells that are constantly active. Well, some cells would never move.
Right.
And you wouldn't want that. So for the moment, I would assume these are all very similar to scale, and that they define some sort of space. The dimensionality of the space is determined. It depends. We'll see how that's determined in a moment. But in some sense, the scale of the space is going to be the same for all spaces that are represented by this thing. I'll talk about how you can stretch it out, not stretch it out. So the point is, though, you've got a bunch of one-dimensional bits on modules, assuming they're all at relatively the same scale, but they're going to as you move, they're going to update depending on what your orientation is. And what I'm going to say here is that that each mini column, here, let me just walk through what I want to bumps, movement, set. Okay, so I'm going to argue that we're going to first focus on how these bumps move as based on our sensation, not based on a reference copy mode command. Okay, I'm going to explain how that works in a moment. And how fast they move depends on the orientation of the sensor and the speed at which it's moving. If I move faster, then these things have to move faster. If I move slower, they're going to move slower. And which ones move depends on which way my orientation is. So therefore, there must be a mini column by mini column scalar that represents the current orientation of that mini column relative to the dimension, to this dimension. So it's like, again, there has to be some sort of scalar which is sort of represented here, but showing how much aligned this dimension is with the current orientation. And that there could be anywhere from no movement to some movement to a lot of movement here, and then that would change this. What I'm going to argue now is that this function here is going to be sensed. Now, I'm going to talk about sensory input. I'm going to have to ask you to think about sensory input different than we typically think about sensory input. Well, I'm going to talk about, if we think about our sensors, there are two basic types of sensors on our skin and our retinas. There are ones that represent spatial patterns and the ones that are directionally sensitive. So on the skin, there are sensors which detect movement in particular directions, and I believe there are in the eye as well. And those are separate channels that go to the cortex. There's these movement directional sensitive things, and there are spatial patterns. Like in the fingers, there'll be some receptors that detect depression, and others detect movement across the skin.
It's the same in the retina.
Yes. Yeah. So I'm saying let's just focus on the movement sensitive ones. And I'm going to argue that those movement sensitive ones are going to basically solve all this problem. So imagine I have this sensory input, but it's the movement sensitive one. So now think about it. I have a bunch of axons coming in, and they just represent movements in some direction on some sensor. We don't know what they are. So what we're going to do is we're going to put them through a spatial polar-like process, just like we always do. Imagine these are going onto some bipolar cells. For the moment, it's like our spatial polar. And it's going to take all these different possible directional sensitive bits, and it's going to divide them up into a nice set that sort of spans the input space, like the spatial polar does. But the outputs of this are going to essentially be directions. They're going to be essentially 1D directions in space, meaning I'm going to look at all these things and try to divide up all these different directional bits. And if it turns out to be that I can only move the 2 dimensions, then I'll end up with a set of mini columns that are faced like this. And if I have 3 dimensions, I'll have a set of mini columns that are faced all over the place like this. This took me a little while to get my head around. Maybe it's easy to understand, maybe it's not. I don't know, it's hard for me. But the idea that we can, instead of just taking spatial patterns, which is what we typically think of, we take these directionally sensitive things, which don't really care what the physical pattern is. It just says which way is the pattern moving on here. It doesn't matter if it's dots or lines or edges, it's which way is it moving. And if you do that, and you do exactly what we always do, you'll end up with columns that represent 1D directions in space. And it'll divide up whatever the space is into the right set of things. So you can think of it down here. I'm going to go down to this picture down here. And now this is very, very speculative, but I'm just giving you a sense of how this might work. We know that there are two basic inputs to every column. There's the inputs to the upper layer, which we tend to think of from layer 1, layer 4. But we also know there's a sensory input to layer 5A. That's well documented. And no one knows the makeup of it. We talked about it, we don't know the makeup of it. So just hypothetical. Imagine that this is actually the sensory inputs that represent direction of movement, not pattern. And imagine I'm feeding into a bunch of bipolar cells here, which then define mini columns. I'm restricting this purely to the bottom half of the layers of the cortex. And so now I've defined a set of, by doing this with the spatial ruler-like function, I will then define mini columns here that are one-dimensional directions. And the activities of these cells, meaning how active they are at any point in time, is exactly this. So if I'm sensing movement, then these cells will become active in a graded fashion, and that graded activity would be the signal I would need to know how to move these cells. They would just, that's the, I don't have a proposal of how it happens. I'm just saying, but that is the signal I want. This is in vision, of course, we can, I think the way this might work in vision, I'm struggling a little bit in touch because I don't know how I get 3D out of it. But in vision, if I have these directionally sensitive bits coming from the retina, if I have ones that are converging or coming apart, I would move in 3 dimensions like this. If they're all moving this way, then I know I'm moving this way. So I think that would work. I don't know how to get 3D out of touch yet on this one. But anyway, so we're basically, I'm saying, okay, what this defines, if I do this, if I took the directionally sensitive sensory bits, I put it into a spatial ruler-like process, define mini columns, and in that activity of those cells move these bumps up and down, I have a metric space with location and orientation. It solves a bunch of these problems. It's interesting, I'll just throw in a couple other things before I get to my things I'm confused about. Notice that if I have, we know that every column has a motor output. There's these layer 5B cells, which are motor output. So we assume they're in every mini column. One way to interpret this is that this, I'm not even explaining how it might get generated, but the way you interpret that is, if I had a layer 5B cell here and it becomes active, then that would be movement in this, it would be a scalar of how much I want to move in this direction. So if I want to move in this direction, some of those layer 5 cells would be very active, some would be less active, some would be less active, some would be not active at all. So the inverse of the input. So for the first time, I had a way of imagining how I could take an output of a column and it would represent direction in n dimensions, like how I want to move my sensor right now in n dimensions. So that's a nice thing that came out of that.
Please like the video,
I'm
folks.
really speculative here. So this only defines the metric space and the location and the orientation in that metric space. I still have to learn an object in that metric space. I have to assign some feature to some location. And we've always thought about that as, the projections back and forth between layer 6 and layer 4, which are built.
That would still work here.
It would, but this has to be something different though.
Right.
Right. So I'm saying here, this has to be a spatial pattern. Like here I was recognizing a directional pattern. Here I need to say what is the spatial pattern that I want to associate with that point in location space. Does that make sense or no?
But those are also directionally selective.
Well, layer 4 cells less so, right?
I think that's one of the tricky things about this, the fact that your requirement is for these bipolar cells to be to only work in the infragranular layers.
No, I didn't say that actually.
Okay.
I didn't say that yet.
But one sort of interesting nugget about that, I think this is true with bipolar cells. You have a whole bunch that are where the cell bodies at layer 2, 3, and they go down, and then you have another set where the cell bodies are in layer 6 and go up.
That would be perfect, because what I'm
that gives you like two.
Yes, what I'm going to propose here is we have 2 basic copies of this basic thing going on here. Then the upper layers, we want to build a feature model, if you will, place cells. And so these spatial things would come in, and they would just, I don't know yet if there's two sets of mini columns and they're intersecting or somehow we're uniting them. I don't really know. But we would put these into the bipolar cells up here, a different set. I'm only suggesting bipolar cells because we need a way of forcing a bunch of cells into mini columns.
Yeah, we need some competition. Like, okay, we're gonna take all.
Yeah, and I need the individual cells in here
to be the same.
Right.
So so putting in the bipolar cells up here, which now if I use a spatial pattern here, so these are bits that would come from the other part of the sensor bits that are not directly sensitive, but more centered around or spatial. Then I would be this goes back to our sort of our current spatial puller and so on, and I would end up with a spatial pattern here that's not directionally sensitive, but it really matters what the individual arrangements of bits are. And so it's very hand wavy at this point in time. But I would say if I sense spatial patterns up here and I sense directional patterns down here, what I think I can show is you can build the entire metric space and orientation, all that stuff down here, and it'll look like what we see in the cortex. We see responses like this across many columns, but we think of it like, oh, it's an orientation. No, it's actually not. What it really is saying is this is going to represent a movement in this direction. And when people try to characterize cells in the cortex, what they do is they give these moving gradients, right? These moving swing waves and so on. Because that's what gets those cells to respond. So that's what I'm arguing is happening here. They're really defining the dimensionality of the space, not the pattern that you're observing. But I do need to observe the pattern, and so I'm going to do that in upper layers. And it's all hand waving, but the idea that you would have two sets of bipolar cells, and I don't really know how they work together. That's the question. So. Then we can go down over here and say, okay, so where are the bits on modules in a column? I'm arguing that this is just a made-up hypothesis, that they're in each individual mini column. We need many of them. We will have hundreds of them in any mini column. What is the dimensionality of bits on modules? Well, it's 1D, and where are the orientation cells? I'm arguing the orientation is actually encoded in the mini columns themselves. Through the 5A input there. How do the orientation cells and grid cells work together to be more to move active grid cells correctly? I've always wondered this, like how do we move, which way to move the pump based on orientation? Well, it's very simple in this case. We just have the scalar. That scalar just says when I have activity in this cell, this input cell here, which is doing this, it's that when I have activity there, the amount of that activity is how quickly I move this.
And each mini column is independent.
Each mini column is independent.
Yeah. Just with 1D. And just to get this straight, right? It's essentially combining the idea of the Lucas paper,
yes.
Wait for reducing the dimensionality of an n-dimensional space. So which could be like a 2D or moving around, a classical grid cell by cell thing, but it could be any conceptual space, and you're using them one-dimensional.
When Marcus and Mirko worked on that paper, I immediately jumped to the idea like, why wouldn't this be done in one-dimensional modules? That would be the most efficient way of doing this.
Right.
And so I'm going to come back to that in a second here, but it's the same idea. So
right. It's essentially merging that idea with mini columns.
Yes, right. So I answered that question. I don't know if I've answered this question. I think I have, but I don't know that. How do grid cells and orientation cells get anchored to the sense object? What I've argued is that I'm not sure if I've done that yet or not. I think it might involve this interaction. So I think I've answered these questions.
And why is that any different from what we did before?
I'm not sure it is. It probably isn't.
I don't think it is. I mean, we just assume 2D and now it's 1D.
Yeah.
The same properties apply.
Yeah, I guess it requires that you don't know you're anchoring. You don't know you're anchoring until you sense something.
Yeah, but that's the same previously too.
Yeah, right. That's right. So
you still need to be able to unify across the different directionalities in the supergranular layers through lateral connections to make it because the problem is the mini columns now divide up the different orientations. And so that if that is true across the column, then in order to link the grid cells with the sensed object, you somehow need to have lateral connections to bring them together.
No, you don't need lateral, but you get a whole bunch of them.
Yeah, you have a bunch of active cells up here and a bunch of active cells up here. And all that matters is that this mass projects up here and this mass projects down here. That's all that matters. So I don't need connections here. I just need that when any particular, if I want to do an associated learning between the sense and the location, I have some cell here and it just randomly subsamples from up here.
Well, yeah,
Yeah,
so
that's
I mean,
what
for Florian's sake, the top one would have a dendritic segment, which gets all of the green ones from the left.
Right, so there's some convergence or something.
There's a convergence.
That's what I meant. You somehow have to, because the mini-columns break this apart into separate modules. So you somehow need to bring it together. That would be one way of
That's a
doing it.
convergence. So you don't need lateral connections, but
It's the lateral connection.
it's convergence from below to above.
They're lateral only after you pass from top to bottom, bottom to top.
Yeah, right.
They're not lateral in these areas here.
Right.
Okay. So that's always been a requirement of our model, and it works beautifully here. But now, since I have so many of these grid cell modules, I really can just do a very casual subsampling of them and just guarantee the work. Okay, I have some questions here, and I'm almost done. First of all, is there any evidence that layer 6 cells behave like this? This is a very unusual proposition that there's a bump in layer 6 that goes up and down. I've never seen anything like that, so that's an interesting question. It's certainly speculative. Could grid cells in the entorhinal cortex also be 1D but look 2D? This is an interesting one. So let me explain this. We've looked at the grid cell modules in entorhinal cortex. Everyone describes them as these 2D bumps moving around. But we've also known that you have to have multiple grid cell modules to do anything useful. And there's no evidence that we've been able to uncover from the published research that there's a whole bunch of grid cell modules of the same scale coinciding or near each other or anything like that. It's like, oh no, we have this one 2D module here, and then we have another 2D module at a different scale, and we have another 2D module at a different scale. And that never really worked out. It doesn't really make sense. We need multiple modules. We have to write a lot of modules at the same scale. So I'm wondering, could it be that the grid cell modules of the entorhinal cortex are also one-dimensional, but they look two-dimensional based on the experimental paradigm that's been used?
So, I feel like this is where Marcus would be helpful. I think that question has been explored.
Is that right?
I think so.
Oh, I didn't know.
Marcus would know that.
Well, let's mark it down for Marcus. The other question, so the alternate is that the alternate would be that no, in the entorhinal cortex, it really evolved to be a two-dimensional world, a two-dimensional space. And that's what it's designed to do. And anytime you try to get away from 2 dimensions, it's going to do some tricks. But then your critics came along and said, no, I'm going to do it completely different. I'm going to do it with one-dimensional modules. So that's a possibility. But it is also a possibility that the grid cells themselves are really one-dimensional. And I have a need for them to be like that, which in this case would be very interesting to ask this question. Is there some way that we can think about where this sort of one-dimensional module, lots of them together, looks like the 2D modules that people measure? And maybe there's evidence to that. I didn't know that. But there's a lot of weird things about the 2D module. Remember the tank paper, which showed that you had multiple bumps moving around, but the activity of the bumps changed over time. It's like, whoa, this is weird, So I think these are interesting questions to ask here.
But you could get that through some kind of inhibitory dynamic, I would presume. But then you would, of course, have to answer the question whether it's something.
Well, why would this look like a 2D grid cell module? Is the question.
Right.
If, for example, maybe if I'm looking at only maybe my imaging technique is I'm looking at one slice like this, and maybe in this one slice it looks like 2D, but I'm not really seeing the whole thing.
And when people do single cell recordings.
Yeah, but single cell recordings, you're sticking a probe in, and it's really hard to tell that when you're aligned with mini columns. It's almost essentially impossible to tell.
I don't know. You're right.
You have to do it afterwards with histology or something.
This is a very speculative proposal because I have no evidence for it. But logically, it solves so many problems.
Plus, it's mathematically beautiful, in the same way that the glucose paper is beautiful.
So, to me, I think it's less mathematical in my mind. It's more sort of biological.
Right. But you mean the biological observations of all the orientation selectivity by mini columns?
There's all these things that have bothered me forever about these are all.
I don't think I've ever heard you say over complete basis set before.
No.
That's good. That's pretty mathematical.
Well, but don't ask me why an equation for it. Okay,
That's a concept.
I know.
Concepts are right. It's just, I don't want to think about it as an equation. Yeah, I learned that in college. So
Different question. I mean, at this
time,
maybe
4 minutes.
maybe it's almost a math question, but why 100, 120, 150 mini columns per macro column then? If the idea is that you get the 1D like wise split up in the mini columns, and so you're using it because your conceptual space, if it's actual spatial 2D, but it might be a lot more high dimensional.
Okay, it's n-dimensional, say you have 4 dimensions, but I still might have 400 mini.
Exactly. So like maybe even in conceptual space, things might be way more complex than that.
So why have so many?
Yeah, why have so many? Right?
So I can think of several reasons. First of all, remember we need lots of mini columns because we need lots of Bitzel modules to just all to do with the fact of sparsity in large spaces, right? We need a lot so we can subsample and still get reliable results. We need a lot so that we're not any one of these that doesn't have to be very particular accurate, right? As long as I just picked 20 of them, that's going to be good enough. But if I had like if I was doing a 3-dimensional spaces and I had 3 mini columns, well, technically it can work, but it's not going to work from a biological point of view, right? Those cells have to be super precise. They would have to all be perfectly, orthogonal, and they wouldn't have to be perfectly orthogonal. But then I'd have to accurately read out the 3 values just so. By doing this, I get sort of a mass, population code, and all the benefits of sparsity of population codes.
There's also this issue of unions. I don't know if
I didn't even talk about that.
Yeah, Florian has some pretty interesting proposals on that.
But,
but if you just think about a pure union, then you need more.
Yeah,
it's better to have more.
So this to me, this is why I called it an idea. I think this is like the basis of a set of investigations. It's not the answer. It's a basis of the set of investigations. It's a way of saying, hey, what if this was like this? How would we start thinking about all those other things again?
Right.
Including things like our what's the name of the other types of cells we postulated, the ones that are displacement cells,
right.
Like displacement cells and different states of the object and so on. We have a lot of other things I didn't mention here that have to be overlaid on top of this idea. There's also other physical things we talked about, like in the ice cube block. Why does it in visual cortex? Why does it look like we have slabs? Is that really what's going on? Yes, there's physical things we haven't talked about.
Right.
There's other requirements we haven't talked about. But what I'm proposing is what really excites me about this is I have a solution for how I get many grid cell modules, how orientation works, how I can derive that from sensory input, which is the key thing. This idea that you do a spatial pooler over directionally sensitive motion detectors, and it will detect, it solves all those problems at once. It basically collapses all these problems into a simple solution, and I've never had a solution for any of it. So that to me what's just like exciting about it. Oh, we talked about one more thing here. I still have this idea that the theta rhythm that's between the thalamus and the cortex could be as a way of scaling things. We've talked about in the past. So he asked about scaling. That this idea that this rhythm here could be a way of accelerating or deaccelerating the movement of the bumps, independent of what you're sensing, right? So that was like the vision thing: bigger and larger, small coffee cups, bigger.
I mean, one thing that is known is that there's a very strong link between, at least in rodents, there's all this data showing that there's, I don't know like precise details, but there's this correlation between whether the animal is moving or not, and has a very strong impact on theta power. I don't quite recall in what way and if that was like layer-wise specified, but I do remember that there's a very strong link between theta power and whether the rodent is moving or not.
Yeah.
So as soon as the rodent stops, there is an abrupt change in theta.
So I think the thing I thought about that, I think I was aware of that. The idea here is that we can update our location by sensory input. Playing the video game and seeing things moving around like this, or if it's feeling my finger moving like this. But then I want to be able to learn to correlate that movement with some motor behavior in the world. And I think the theta signal is sort of when my current thinking in it's very spectral, it's one logic you just said there. It's like saying, okay, I'm either moving and how fast I'm moving and not moving, but it doesn't have to give you directionality. It just says you're moving. And that could then update these guys based on your current orientation as well. So it could be like saying, okay, I'm not going to update these guys because you're moving. I'm not even sensing anything. I got my eyes closed. And so as I do that, this thing comes up and down. So there's two ways of moving these guys. One is that you have this sense thing. There's a flow of visual field. You're feeling these things moving against your finger. And the other way is that, oh no, I'm not using that. Or in addition to that, I'm detecting physical movement of the thing, and I'm going to update these based on that.
So I'm a little confused how motor input plays into this. Direction sense is a passive thing. Well, I guess in the sense of touch, they're tied together. But for vision, the direction selectivity would be passive. It's the object is moving across a sensor, as opposed to I'm moving.
No, I don't think it has to be.
It doesn't have to be passive, but it can be passive.
But I can't I just, even if it wouldn't even microscope cards make this work. I have to think about it.
But it's not a direct movement sense. Right, if something is moving across my field of view, it's going to cause that direction to light up.
Okay, I wasn't even talking about that. All right, so I haven't even dealt with that issue. I'm talking about how I'm sensing static objects at the moment. I haven't talked about motion of an object. That's a separate question. So I'm abandoning that idea. I'm just saying, imagine I'm just sensing a static object.
But you say isn't there also a motion command in here somewhere?
When you say motion command,
like a reference copy of movement.
What I'm suggesting is, I haven't really worked it out, so just leave it. I'm suggesting that the way to start thinking about this first is really focus on this one. I think this is the primary way we learn the dimensionality space. This is the primary way we actually detect motion. A secondary way has to be learned. So there's going to be some motor command coming in, but a piece of that could be just the theta signal. And if I knew my orientation already, if I already had my orientation set up, then all I need is to say, how fast are you moving? And that would be sufficient, because then all these guys would update correctly, independently, based on my orientation. So I don't have to pass in on this speculative miniature version of it right now. I'm saying I can imagine motion being reference copy really is it's mostly just knowing, am I moving? And then somehow I have to know what is my current orientation. If I know my current orientation and these cells somehow represent my current orientation, then this movement theta will just update these things, and that's it. I don't need anything else. I don't need to know.
So all the many comes together already is like sort of like establish this vector in an n-dimensional space.
Yeah.
And all you then need to know is the magnitude, like in polar coordinates.
Yeah, basically. Right. I just need to know. I already know if my mini column knows its projection onto the overall orientation. Then all I need to know is how fast I'm moving that. How fast am I moving? And I'll just pick out the component vector of that.
Is there a model for how the theta wave is generated?
Yeah.
There is.
Yes.
And we have to stop. I promise not to stop.
Yeah.
So we're going to end this right now. Thanks, Matt. We have a meeting.
All right. Thank you. I'm going to turn this off now.
Of how grid cells work.
Let me turn this. Hey, can one of you guys take the double back for me, please? Okay. All right. Hey, that was exciting, huh? Okay, so I'm going to have to rewatch that because I was distracted and it seemed like really interesting stuff. There's a lot of theory here that Jeff was talking about that I haven't heard before. So this is the first time we've thought about it, first time I've thought about it. So if you want to discuss it, the forum link is in the video description. If it's not, I'm going to put it there right now. Thanks everybody for chatting along. I'm busy, as you can see. My office is a mess. I have to get to work now. But stay tuned for more research videos Probably another one next week, at least one, maybe one this Wednesday too. Anyway, chat. Goal sparse representation theories derive a dictionary of atomic features that can be linearly combined into any observable feature using a few atoms as possible. That's a good thought. Falco says, great stream. Thank you. Oh, guys and gals, folks in general, please like the video and subscribe to our YouTube channel if you haven't already. Good number of people watching. Thanks again for watching. And let people know what we're doing here at Numenta. This is very unusual in the science community to be, I think, live streaming a private company's research meetings in the realm of artificial intelligence. You're getting definitely bleeding edge stuff here from Numenta as it's coming from our founder's mouth. This is the first I've heard of this theory, so interesting stuff. John was asking, do you think Jeff has the same intuition that understanding spatialness as a basis vector trivia cases will lead to extrapolation to concept math? Absolutely, he has an intuition. We've talked about it many times. That is the idea. That's where we're trying to get to, understanding thoughts as things that can be computed and compared and et cetera internally within an agent system. Now, how useful those representations are outside of that system is debatable, and we've had those debates when we've talked about agency and knowledge transfer in the past on my Twitch stream. I think you can find those on our YouTube channel if you search for agency and knowledge transfer as well. What happened to I messed the chat up here. Hold on a sec. I can't see where the chat is. There we go. Okay, you're welcome. Yeah, I'll be rewatching this too, so don't feel bad. Thanks again for watching, everybody. I am going to cut the stream short. New HTM school this Friday. Promise. It'll be good too. Bing. Take care.